you know, I speculate.
Okay, thank you. Any question? Then, thank you again, Angel. We will move forward. Our next and last speaker is María Llorens-Martín. She's currently a senior researcher, tenure scientific at the Center for Molecular Biology, Severo, Ochoa, in Madrid, and also she's a member and PI of the CIBERNED. Today we could enjoy the talk of the Joint Research Recognition that was awarded for one of the members of Maria's lab. And as you know, her research focused on the biology of the newborn granular neurons and the neuroprotective potential of the adult hippocampal neurogenesis for the treatment of neurodegenerative diseases. Her talk today is about adult hippocampal neurogenesis and neurodegenerative diseases that we have pinch this morning in the award. Okay, Maria, this is getting ready.
Apologies for it worked before, but
software y ya está.
Have a little problem with the computer, the change the computer, but we are going to fix it. Okay. Yes. The good thing always we have to wait.
Tres, cuatro, nueve, 349 967, no es mi teléfono, ¿eh? 152. Okay, here we are. Okay, so thank you very much. First, to the scientific committee for the great honor of closing this excellent summit. Thank you all for staying here until the very last moment. And thank you for your help. So I would like to apologize because some of the data I will present have been already presented by Julia in her talk, but I hope that our two talks will complement each other. So adult hippocampal neurogenesis encompasses the generation of new functional neurons in the adult brain. It provides an extraordinary reserve of lifetime plasticity to the adult brain. As Julia mentioned, adult neurogenesis is spatially restricted. It only takes place in the so-called neurogenic niches. And today I will talk about the subgranular zone of the hippocampus. This neurogenic region is important for learning, memory, and the regulation of mood and emotions. Also, the hippocampus, and in particular, adult neurogenesis, are dysregulated in neurodegenerative diseases and also during physiological aging. This is a reconstruction of how the life of a new neuron looks like in a rodent. You can see how the life of these neurons starts with the asymmetric cell division from neural stem cells that are resident in this region, in the subgranular zone. It continues with subsequent differentiation stages during which these neurons acquire a progressively more complex morphology and finally conclude with the synaptic integration of these neurons in the hippocampal circuitry. The classical way in which adult neurogenesis has been studied in rodents is by using specific cell markers that characterize each of these stages and permits an accurate classification of these cells throughout their life. Adult neurogenesis has been demonstrated to occur in 121 mammalian species, but somehow the question on whether our human beings are unique has been sounded in the field for the last decades. So this is a summary, which of course you cannot see, but it's published of the works that have been addressed the occurrence of adult neurogenesis in the past and presently. And an enormous controversy was raised in the field a couple of years or 3 years ago due to the publication of several works that did not find evidence for markers of adult neurogenesis in adult subjects. So we have been working on this topic for several years, and when this controversy arose, we thought that probably methodological aspects that were different between these works could underlay or partially underlay those discrepancies. So as Julia mentioned, the study of adult neurogenesis and probably any other process in the human brain is a little bit more complicated than studying the same process in mice. And there are distinct decisions that one has to make when deciding how to process a sample that, as we demonstrated, have important implications for the final results. So we focused our attention on determining whether fixation can limit our capacity to visualize markers of adult neurogenesis in the human brain. To answer this simple question, we used a very simple experimental design with the help of Alberto Rábano, which was to obtain the whole hippocampus from several neurologically healthy subjects and divide the hippocampi into different fragments, each of which was fixed for a different period of time. In that way, we could quantify the number of cells that were positive for the gold standard marker of immature neurons, double cortin, in each of those fragments of the hippocampus. So apparently, one would expect that the number of cells would always be the same. But as we demonstrated, that was not the case. So we were able to detect this specific staining of adult-born or immature neurons with fixation shorter than 12 hours, which, as you all know, it's an extremely short fixation time. After 12 more hours, just after 24 hours of fixation in PFA, this beautiful staining completely disappeared and was replaced by this in a specific background that almost impeded us to visualize cells positive for not only double cortin, but many other markers of adult neurogenesis. So luckily, as Julia told you, we were able to design a very simple histological procedure that allowed us to recover the capability of these antibodies to recognize the antigens in these samples that were fixed for just 24 or 48 hours, which is still a very short fixation time. So our works demonstrate, and this is something that was known since the 60s, that fixation critically limits our capacity to visualize some antigens in the tissue, and in our case, long fixation impeded the visualization of most adult hippocampal neurogenesis markers. Well, this is important because as far as I know, neurogenesis has not been questioned in neonates, right? So even in these samples, which are full of immature neurons, if we fix them for 6 months, staining with immature neuron markers completely disappeared. So, as I said, we consider that this is important because we could be wrongly interpreting this absence of staining as a lack of neurogenesis, which, at least in this neonate case, is not the case. So it is not surprising that an enormous controversy was in the field, given that among the 53 studies that we have found addressing the occurrence of adult hippocampal neurogenesis in humans, 30 of these studies did not disclose the fixation protocol, and 12 of them used really long fixation times. This is important because all the studies that did not find evidences for adult neurogenesis used longer fixation times or used harsh antigen retrieval protocols, which, as we also demonstrated, provokes the appearance of inspecific signal. So once we showed the protocol that worked best in our hands to detect these cells, the next question was addressed to us. So whether these immature neurons that we detected are some kind of forever young neurons. So how can we be sure that they are generated during adult life? Well, we cannot be sure with our data, but it was demonstrated several decades ago that BRDU is incorporated selectively in the human dental jars. Moreover, we provided evidence that these cells are not all equal. So if these cells were just an immature population of cells that lasted forever in the dental jars, one would expect that they were all the same. So, as Julia told you, we found a variety of markers characteristic of distinct maturation stages that were co-expressed by these immature neurons. And moreover, the cells that are expressing more immature markers show immature morphological features, whether others that are supposedly at more advanced stages of differentiation also express a more mature phenotype and morphology. So these data indicate that the process of maturation of these immature neurons is somehow dynamic. However, something that was asked in the field was whether we were not able to detect neural stem cells in our first published paper in 2019. Well, the detection of neural stem cells is even more difficult than that of immature neurons, and it is necessary to use a mild detergent to detect cells positive for nestin, SOX2, bimentin, and other neural stem cell markers. But are they actually neural stem cells, or are they just astrocytes? This is an important question, and we were happy to address in our last paper. So when we stained with bimentin, one general marker for neural stem cells, we could detect the presence of two distinct populations of cells. You can see how some of these cells that are very small and are located in the molecular layer also express astrocytic markers, such as S100 beta. But there is a small population of cells that are bimentin, nestin, SOX2 positive, etc., that lack S100 beta expression. And interestingly, the morphology and the position of these cells is totally different to the former. And these characteristics are shared with those that we know from neural stem cells in other mammalian species. So our data suggests the presence of a population of nestin, bimentin, SOX2 positive S100 beta negative radial glia-like cells that express phenotypic characteristics of neural stem cells. But there is one step missing, which is the proliferation. So how can these cells give rise to new neurons? We also detected the presence of proliferative cells in the dental jars, as you can see here, with distinct markers. But how can we be sure that they belong to the neuronal trajectory? Why don't we say that they are glial cells? Okay, 90% of these cells express also the neuroblast marker, double cortin, and are localized in the subgranular zone. So all these features are extremely similar to those that we know from the rodent brain. So our data demonstrating the presence of immature neurons, radial glia-like cells, and proliferative markers, together with all the robust previous evidence published in the field, support the notion that adult neurogenesis in the human brain is a robust process that takes place until the 9th decade of life. However, as has also been suggested before, the rate of adult neurogenesis decreases with age. And this raised an important question. If this mild decrease is observed during physiological aging, what happens when neurodegeneration attacks the brain? And there are many diseases that we have been learning these days that would be interesting to study, but we decided to start with Alzheimer's disease in collaboration with Alberto again. And more recently, we have addressed the question of whether adult neurogenesis is altered in other neurodegenerative diseases that do not target the hippocampus directly, but can be related to hippocampal malfunctioning. We have studied patients with ALS, Huntington's disease, Parkinson's disease, dementia with Lewy bodies, and with frontotemporal dementia. Throughout the last, I would say, 12 years, we have collected prospectively a collection of more than 100 human brain samples to study adult hippocampal neurogenesis. It was not an easy path, I would say, but it was really exciting to collect these samples together with Alberto. So we first studied adult neurogenesis in patients with Alzheimer's disease that were distributed among the 6 neuropathological stages of the disease. And we first quantified the number of double-cortin positive immature neurons. To our surprise, we detected a 30% decrease in the number of immature neurons in patients at Brach-Tau stage 1, which, as you know, is most likely prodromic, so this decrease in the number of immature neurons probably precedes the appearance of cognitive alterations. Importantly, this decrease or these impairments in adult neurogenesis are independent to the preservation of the whole dentate gyrus. As you can see, the number of new and positive neurons remains unchanged throughout the progression of the disease. This data can be important because the group of Orly Lazarov also demonstrated that the decrease in adult neurogenesis parallels the cognitive impairments in Alzheimer's disease patients. What we found at that time was that the maturation of immature neurons was also altered. So it was not only that there were fewer cells, but the ones that somehow survived also had an impaired maturation. They did not express terminal differentiation markers. And we have also recently, we are currently, we have addressed the morphological differences that are present in these neurons. These are not specifically adult-born neurons, but what Berenice, which is a brilliant postdoc in my lab, has demonstrated is that there is a progressive, a sequence of unusual structural plasticity that takes place in granule neurons in human patients with Alzheimer's disease. So regarding the other neurodegenerative diseases, Julia did a great job showing you the main results of the paper, and I will just focus on showing you that the scenario is totally different to what we observed in patients with Alzheimer's disease, as the number of immature neurons was increased in some of these diseases or remained unchanged. But what was a constant when we compared all the diseases that we studied was the alteration in the morphology of the cells. So most of the cells exhibited more than one primary apical dendrite and an aberrant positioning in the granule cell layer, which somehow we know from rodent data that this can be related to the altered maturation of these cells. Together with the disbalance in the quiescence proliferation of neural stem cells, led us to conclude that adult neurogenesis is differentially, but in all the cases, vulnerable to distinct neurodegenerative diseases. And this also led us to investigate whether the dentate gyrus neurogenic niche, the structure that supports, that gives trophic support to these neurons for their survival and integration, was altered in these patients. As Julia told you, the dentate gyrus neurogenic niche in humans is also composed of vasculature, astrocytes, and microglia, which all together form a complex network that somehow orchestrates or regulates adult neurogenesis. We found that the role of microglia phagocytosis could be important, as it is during rodent adult hippocampal neurogenesis. And as you can see here, we found an interesting correlation between the morphology and the putative function, phagocytic function of these microglial cells, which number is not altered during aging. But in contrast, the phagocytic capacity of these cells measured as the percentage of pignotic cells that are engulfed by microglial cells is dramatically reduced, not only during physiological aging, but also during neurodegeneration. So just as a very brief summary of the alterations in the niche that Julia also summarized, we found in all the diseases a marked astrogliosis of the subgranular zone, which was accompanied by a reduction in the phagocytic capacity of the microglia, which is related to the presence of an increased number of pignotic cells and the astrogliosis in the niche. Moreover, as we were discussing before, we also found an impairment or an alteration in the morphology of the blood vessels in all these diseases. And this can be important because it could be a shared mechanisms between distinct pathologies that are neuropathologically and clinically very diverse, but could be a potential therapeutic target. So, as a summary, apoptosis is a general hallmark of all neurodegenerative diseases, apoptosis specifically in the subgranular zone of the dentate gyrus, also the alteration in the dentate gyrus neurogenic niche homeostasis, but distinct diseases trigger the appearance of disease-associated adult hippocampal neurogenesis signatures that that show that distinct subpopulations of the adult neurogenesis process have distinct susceptibility to the distinct neurodegenerative diseases, the mechanisms that are taking place in the brain. So, as a summary, our work showed the presence of neural stem cells, proliferative cells, and immature neurons in the adult human dentate gyrus until at least the 10th decade of life. Alzheimer's disease impairs in a very dramatic way adult hippocampal neurogenesis, and other neurodegenerative diseases differentially target specific aspects of adult neurogenesis and the dentate gyrus neurogenic niche homeostasis. And also the most important slide of my presentation, which is my wonderful group at the CVM of Madrid. I would like to thank our collaborators, especially Alberto and Jesus, for their support during all these years, and of course our sponsors. So thank you very much for your attention.
Thank you, Maria. Well, we are open for debate or any question from the audience. Okay, Carlos.
Beautiful, Maria. Yes, a question. Can you speculate whether this decrease in the capacity of neurogenesis may be secondary to macrogliosis and astrogliosis?
Well, that's a very important question, whether these alterations are a cause or a consequence of the alteration in the niche. We don't have an answer to that, but given that we observe the alteration in the niche in all the diseases, we believe that this occurs first. And I would not dare to say who comes first, whether microglia or the vasculature or astrocytes. In my personal opinion, probably the vasculature is playing an important role in amplifying the neurodegeneration or neuroinflammation that is taking place in other brain regions.
Thank you, Maria. Beautiful. Do you think in these conditions where neurogenesis is increased in the hippocampus, we should be looking for some signature of this on volumetric MRI?
Well, I don't know why we are not looking at that yet. I think that's the future, of course, and it was attempted in the past, not only by functional magnetic spectroscopy, but also by PET ligands. And I think that will be the future, meaning that would allow us to detect changes in adult neurogenesis a lot of time before the appearance of clinical symptomatology. So I think that endorses an important therapeutic potential.
Okay, great. Because I think some of the samples you've used, we may have MRI pre-mortem for them, so we can look at it specifically.
Yes, pre-mortem, of course, that would be important. I think that the correlation that we could establish in post-mortem tissue would also be important to try to design strategies to find them pre-mortem. But the highest utility would be to detect them pre-mortem. So we have to talk.
Okay, Maria. Somebody? You want to ask?
Very nice, Maria. Have you tried to correlate the findings that you have in these good conditions with what was reported with C14, carbon C14 dating, to see whether the extent of neurogenesis is similarly detected with both approaches?
Well, we have been talking to Jonas about this correlation, and we think that the way in which we have reported the total number of cells is not compatible with that correlation, because we have not been able to report the total number of cells. We report cell densities. So what we did was to delineate the granule cell layer, count the cells that are inside, and then express a cell density. So we would need to know the total volume of the granule cell layer in these subjects to give an absolute number of cells. We have been talking to them about how mathematically this could be inferred, but I think it's not an easy task, and reporting absolute number of cells is relevant and can have important consequences. So I think it's difficult at this time. Maybe with other papers published by Maura Boldrini and Orly Lazarov, who they know the volume of the total granule cell layer, although sampling is a bit different, but it would be easier to correlate. And as far as we have seen in those data, all of them are compatible. Thank you.
I just wanted to ask if that changes you see in the microglia morphology, these changes are of activation because there are neuronal death, or they are degenerative sign of microglia that are not supporting the niche?
Well, what we believe is that means that microglia is impaired in their functionality, because we detected a reduction in the number of phagocytic pouches, which from the data we know in rodents from Amanda Sierra's group, that means that these cells are not being able to phagocytose the cells. And in fact, we found a nice correlation between, in an inverse correlation between the number of phagocytic pouches and the number of pyknotic cells. So we cannot hypothesize which is the role that this microglia is having, for example, on the connectivity of new neurons, but we can suggest that these alterations in the morphology are related to impaired functionality, at least in terms of phagocytosis. Okay.
Thank you. Thank you, Maria, for your beautiful talk and all the speakers. And now we will have the concluding remark from the organizer.